Hello students, in today's class we are going to learn about plasmid vectors, and here we'll specifically discuss about cloning vectors and expression vectors. So what are vectors? These are basically agents that can carry a DNA fragment into the host cell in which it is capable of replication. If the vector is used only for reproducing the foreign DNA fragment, then it is known as a cloning vector. While if it is used for expression or overexpression of a foreign gene, then it is known as an expression vector. So cloning vectors are DNA molecules that are used to transport clone sequences to the biological host from the test tubes. Most of the cloning vectors are genetically engineered and they are used to amplify a single molecule of DNA into several copies. Some of the important properties of cloning vectors include its ability to replicate. They contain genetic marker for selection of the vector. They contain unique restriction endonuclease target sites that can be used for cloning purposes. And they must have minimum amount of non-essential DNA in order to optimize the cloning process. So in general, there are different types of vectors, and different types of vectors can be selected depending on the size of the DNA that you want to clone. For example, a plasmid DNA can accommodate an insert size of about 6 to 12 kilobase pairs, while a bacteriophage genome can accommodate up to 25 kilobase pairs of foreign DNA molecule. Cosmids can accommodate about 35 kilobase pairs, while artificial chromosomes such as the yeast and bacterial artificial chromosomes can take up very large size of the insert ranging from about 200 to 1000 kilobase pairs. So firstly, we'll start with the discussion on plasmid DNA. And so these are used to clone the foreign DNA molecule, which ranges in size from about 100 to 10 kilobase pairs, 100 base pairs to about 10 kilobase pairs. Some of the typical features of the plasmid vectors are mentioned. So first of all, it has a small size. So smaller the size of the vector, the easier will be the transformation process, and hence it is advantageous to have a vector with least amount of non-essential genes. Secondly, origin of replication. So origin of replication, or ORI, is the sequence which is recognized and bound by the cellular DNA replication machinery. And without the origin of replication, DNA cannot be replicated in the cell. Secondly, we have the multiple cloning site, also known as the polylinker region. And this region basically contains collection of unique restriction enzyme target sites. And out of these unique restriction enzyme collection of target sites, we can choose one or two depending on the suitability and use it for cloning purposes. Then the next property is selectable marker. So selectable marker are those markers that are used to ensure that the plasmid vector has been picked up by the cell. So in this case, we have the ampicillin resistant gene, which is the selectable marker that will ensure that the plasmid vector is picked up by the cells. So once we have done the recombination step, that is, we do the ligation with the insert molecule. We first digest the vector and we insert with a restriction enzyme, say, and then we do a ligation reaction. So after we do the ligation reaction, we need to do a transformation step. So a transformation is done by several methods, such as a heat shock method or an electroporation method, for which we first need to prepare competent cells, and then we mix the competent cells with the ligation mixture and we perform a transformation reaction. So after transformation, Some cells will pick up the plasmid and some cells will be empty. They will not pick up the plasmid. Then we provide some incubation time to the cells in a recovery medium. So this is for about 1 hour in at 37 degrees Celsius. So this allows the membranes of the uh, host cells which are being disrupted during the transformation reaction. It provides some recovery time and the medium for the cells to reform their membranes. And after that, the next step is plating on a nutrient medium containing a suitable antibiotic. So in this case, the selection marker for the plasmid is ampicillin. So your medium must contain ampicillin. So only those cells that had picked up the plasmid, they will be able to grow here and form colonies, while the empty cells which have not picked up the plasmid, they are unable to grow in presence of ampicillin. So here, ampicillin functions as a selectable marker for the plasmid. Then we have screenable marker. So screenable marker in this case, in this plasmid, we have laxi gene. Laxi gene encodes for the enzyme beta galactosidase, and the multiple cloning site is actually embedded within the laxi gene. So we all know laxi gene encodes for enzyme beta galactosidase, which can cleave the natural substrate, that is lactose, into glucose and galactose. And it can also cleave the artificial substrate, which is known as X-gal, where the gal part is again galactose, while the X part stands for 5-bromo-4-chloro-free indolin residue. And in presence of an inducer molecule, so the natural inducer is allolactose, while the artificial inducer that we make use of here is known as IPTG, which mimics the structure of allolactose and can be used for induction of lactoserotonin gene. So there are two possibilities here. Either we have a recombinant clone or a recombinant construct in which within the multiple cloning site your insert is ligated, and that is going to disrupt the beta galactosidase gene, or it will become inactive. Or the other case is that you just have an empty plasmid after the transformation and you do not have the clone present within the beta galactosidase gene. And as a result, the beta galactosidase enzyme, active enzyme, will be formed, and that will result in formation of blue colonies in presence or after the cleavage of the substrate. That will release the X part, which is substituted indolin residue. So here we have an image of the blue-white screening where you can see the few colonies are white in color, and these are those in which the beta galactosidase gene is inactive or disrupted because of cloning of the insert, while the others are blue colonies, which are because they don't have the insert and hence the beta galactosidase function is active. So in this way, you can make use of laxi gene as a screenable marker, means you are using it to screen whether your colonies are going to have the clone or not. Then next property is the sequencing primers. So DNA sequencing primers are basically complementary to the regions flanking the multiple cloning sites, so, which are represented by these arrows here. And they are basically used for sequencing reaction after cloning to confirm if the correct insert is cloned or not within the vector. So next we come to the expression vector, and we'll discuss about expression vector with the help of example which is known as the PET15B vector. So some of the properties of the vector are similar to that of the normal cloning vector, such as it contains origin of replication. So origin of replication here is being derived from PBR 322 plasmid. Then we have a multiple cloning site, which is used for cloning. A selectable marker, here we have an ampicillin resistant selectable marker, and sequencing primers, which are similar again to that of the cloning vector with slight modifications. And apart from that, we also have a regulatory region present here, which is encoding a LAC-I gene, which is encoding the repressor protein. So here we have the detailed structure of the important regions within the expression vector. So first of all, we have the T7 promoters here. So T7 promoters are basically promoters from a phage T7. They're derived from phage T7, and these being the strong promoters, your target gene is basically cloned downstream of the T7 promoters. That is going to enhance the production of the recombinant protein, or basically going to increase the expression of the target gene sequence that is being cloned. Then we have the T7 promoter primers and the T7 terminator primers. So we basically have forward and reverse primers that will bind to these regions, and they are used for conformation and sequencing of the clones that are generated or constructed. Then what else? We have a LAC-I coding sequence, as we had seen in the previous slide. It encodes for the repressor protein, which binds to the LAC operator region present downstreams of the T7 promoter, and thus it prevents or inhibits the expression of the clone gene unless required. So basically what we want is that the expression of the foreign gene or the clone gene should take place at particular conditions, such as when a particular cell number is reached, particular incubation conditions such as temperature, pH, media conditions, etc. So normally in an overexpression experiment, we first want to grow the cell number. And once the cell number is grown, then we transfer the cells into a fresh medium, and then we incubate them at the required conditions, which are optimal for protein formation. So that now the cells don't have to divide, they just have to express the protein. So here we make use of the repressor protein. And so repressor protein is basically going to inhibit the expression from the operator. And when we want the expression to take place, that is after subculturing and incubation at the optimal conditions, we want the expression to then start. So at that particular time, we add the inducer molecule, that is the IPTG molecule, which is going to inactivate the repressor and will allow expression of the clone gene. Then multiple cloning sites are present. So we have different multiple cloning sites present within the different restriction enzyme target sites present within the multiple cloning site region, like the ND1, NHE1, BAMH1, EcoR1, and so on. So here in this cloning reaction, we basically want to do a directional cloning because we want the start codon to be present here and the end and the stop codon present at the other end, because otherwise if it is cloned in a reverse direction, then you are basically going to generate a wrong protein. So we need to do a directional cloning when we are using it for protein overexpression. So we basically need to make use of two restriction enzymes. Say for example, the first restriction enzyme is NDE1, which is going to clone the region towards the start codon, then the second restriction enzyme can be any of these which are present downstream, which will be cloned towards the other end, that is towards the stop codon end, for which we make use of the multiple cloning site. Then what else we have? His tag is present here. There are also other tags present, such as the T7 tag. So the importance of the tags, for example, the his tag. So it adds a string of about 6 to 9 histidine residues towards the C terminal end of the protein. Now also for this we need to ensure that when we do the cloning, the gene will naturally contain a stop codon. So we have to design the primers before the stop codon so that the protein synthesis will not stop at the stop codon that is naturally present in the gene. So we have to design the primers accordingly, that we skip the stop codon region. And then as a result, a fusion protein will be generated. So it will not stop at since the stop codon is not there, but it will proceed beyond that and will express the sequence that is present in the vector. And this allows addition of a tag such as the his tag towards the C-terminal end of the recombinant protein. And then there is another stop codon present within the plasmid vector itself that will stop that is towards the end of the his tag, and that will stop the expression of the recombinant protein. So what is the purpose of using these tags? Firstly, they can be useful for easy purification using nickel anti columns. So these columns basically can bind with positively charged tail of the recombinant proteins, which is generated because of the presence of histidine residues. So it can be used for easy purification. And secondly, it can be also used for detection of recombinant protein. So there are these commercially available antibodies for the his tag region. So these antibodies will basically bind and detect the recombinant protein. Otherwise, you need to have antibodies developed for your recombinant protein first. But by making use of rather his tag antibodies, you are basically getting a fusion protein here, your protein and the his tag. And if you already have antibodies towards the his tag regions, then that can be used for detection of the recombinant protein. Then the next slide talks about the different conformations of plasmids. So when we do a plasmid prep reaction, it is important that we isolate the supercoiled form of DNA. The supercoiled form of DNA doesn't has any nicks present in both the strands of a DNA. While there are other forms of DNA, such as you can see the open circle form of DNA, where we have a nick present in one of the strands, there are other forms of plasmids also present where you will have presence of nicks, one nick in both the strands present at different locations, like one may be here, the other may be here in the inner circle, or you can have linear forms or shredded forms, different forms of plasmids can be present, and they basically run at a different position when you observe them in an agarose gel electrophoresis gel. So basically, for a successful cloning reaction, we need to isolate the supercoiled form of DNA. Otherwise, what will happen if the plasmid is already having nicks at multiple ends? When we digest, then you may lose those fragments that are present and you will not get a successful cloning product. So in order to get a successful cloning product, your DNA should not be shredded at any of the locations, and we must ensure that we have isolated supercoiled form of DNA, and only then we proceed with the cloning reaction. So as you can see in this diagram, in this gel picture, we can see that the supercoiled form of DNA is running at a smallest position, and then we have the linear form of DNA. So when we digest the supercoiled form of DNA, it gets converted into the linear form, which runs at a higher position. And then those DNAs that are shredded or having several nicks within one or both strands, they will run further at further higher positions. So a cloning reaction will not work if we are using these two forms of plasmid. If we begin with these two forms of plasmid for a cloning reaction. So hence it is important that we start with supercoiled form of plasmid DNA for cloning reaction. So that's it about plasmid vectors. Thank you very much, and kindly like, share, and subscribe if you like the contents.